Hi, everyone. Welcome to chapter 21. We're going to discuss diseases of the respiratory system in this chapter. So let's get going. Well, I guess we're just going to skip right on through. I guess we could, though. You guys already know what this slide is. It's just going to be the same as all the other ones. We're just talking about these diseases and getting into the details of them, just relating in this chapter to the respiratory tract. So the respiratory tract will be divided into two sections, essentially, the upper and the lower. The upper includes the mouth, nose, nasal cavity, sinuses, throat, which is also called the pharynx, and the epiglottis and the larynx. The lower is the trachea, bronchi, bronchioles, and the alveoli. As far as the anatomical defenses go for the respiratory tract, we have the nasal hairs themselves to trap things, the ciliated epithelium of the trachea and the bronchi that helps move things out, including mucus. Mucus itself counts. We're getting things stuck in that and moving it out by the ciliated epithelium, as well as coughing, which is the next one, sneezing and swallowing, just to clear out everything coming out of the respiratory tract. The second and third line defenses for the respiratory tract include complement action in the lungs, increased levels of cytokines and antimicrobial peptides, alveolar macrophages, and secretory IgA. Sorry, a lot of this stuff is not very exciting, and I know it feels like I'm just reading all this stuff off to you, and I apologize for that, but it's mostly memorization chapters. So moving on to the normal biota of the respiratory tract. The upper tract has quite a large number of commensal organisms living within it. I'm not going to get into the details of that, but we have bacteria, quite a lot of bacteria, and we have yeasts as well. All of these are important in microbial antagonism, which is where the microbes themselves are going to help protect us from the bad microbes that would be coming in. So bacteria that would be considered normal, well, could be considered normal biota for some people, but could potentially cause pathogenic effects include Streptococcus pyogenes, Haemophilus influenzae, Streptococcus pneumoniae, Neisseria meningitidis, and Staphylococcus aureus. We'll get into some of these, and some of these you're already familiar with. First, which portion of our respiratory system would have the least microbes in a healthy individual? Like a lot of other I don't know why I can't talk right now. Like a lot of other systems that we have been talking about, the deeper you go, the less microbes typically will be associated with it. So we're going to go with alveoli. All right. The next, the first kind, I guess, breaking it down disease we're going to get into is the common cold. Common cold is also known as rhinitis. It's inflammation, infection of the nose itself. Okay, so it's pretty costing the U.S. a lot of money, a lot of missed days of work, whatever. Then we have the signs and symptoms associated with it. If you don't know these, I don't know where you're from. Sneezing, scratchy throat, runny nose, typically no fever, and this can predispose people to secondary infections, infection with bacteria that'll be opportunistic due to the inflammation in the area, infection caused by a virus typically causing the cold. There are over 200 different kinds of viruses that can cause the common cold. 99 of those are going to be serotypes of rhinovirus, coronavirus, and adenovirus. There are a lot of coronaviruses out there, not just COVID. I do want you guys to keep that in mind. That's just kind of one kind of coronavirus, okay? Symptoms are usually due to our immune response to the viral infection and not actual virulent factors of the viruses. So just our response is causing the symptoms. Remember, our immune system causes a lot of inflammation when it responds to bad guys. So that's how it gets rid of bad guys, but we also feel the effects of that. Transmitted by droplet contact, so sneezing and coughing and everything like that, as well as indirect transmission that will include things like fomites. Remember that term. I'm not going to actually explain it again because I'm sure you guys know what it means by now. And if you don't, you are more than welcome to look it up. Okay, you should know by now. I'm sure you do. So there's no vaccine for the common cold, and no drug or specific chemotherapeutic agents for the cold. There was an attempt to make a vaccine for about 50 types of different viruses that can cause the cold and was going into human trials. Sinusitis is something that can develop as a sort of secondary infection to colds as well as on its own. This is just a sinus infection. Inflammatory conditions of any of the 4 pairs of sinuses within the skull that can be caused by allergies or infections. And patients that are suffering from a cold also tend to develop sinusitis, we just said when we're talking about the cold viruses. Signs and symptoms of sinusitis include the following: sinus pain, nasal congestion, pressure, headache, toothache. A lot of that is referred pain just from swelling within the skull. And then discharge can be opaque, yellow, or green in color. Typically, that's going to be associated with bacterial and or fungal infections, and the viral infections tend not to have quite as much of that discolored discharge. Next, we have acute otitis media. That is also the ear infection. Signs and symptoms: fullness and pain in the ear, hearing loss. In young children, you can have irritability associated and trouble sleeping, and can cause the eardrum to burst if it gets particularly bad. You can treat it with tubes in the ears to help drainage to relieve the pressure, but typically you're just going to wait it out for about 72 hours to see if it improves. If it gets worse, then you would go to get tubes. All right, next we have pharyngitis. Here we're just talking about inflamed throat, right? Pharyngitis is just inflammation, infection in the throat. This includes strep throat and tonsillitis. Signs and symptoms of pharyngitis might include inflammation of the throat, causing pain and swelling. We'll have inflammatory packets. We'll see white little patches on the throat as a result of that. That's due to the white cells building up in those areas, and that can be associated with difficulty swallowing as well as foul breath from bacterial growth in the back of the throat. Viral versions of the pharyngitis infections can just be mild and cause hoarseness in the throat, whereas the bacterial ones are usually more painful and accompanied by fever, headache, and nausea. That includes strep throat, which is caused by Streptococcus pyogenes. Streptococcus pyogenes, we have talked about it previously. It can be particularly nasty in some cases. It is a gram-positive coccus that grows in chains. It is facultative, anaerobe. It produces capsules and a slime layer, so a lot of virulence factors there. It also has exotoxins that it makes, and more virulence factors that can be associated with pathogenic symptoms. Untreated strep can result in scarlet fever, rheumatic fever, glomerulonephritis, and necrotizing fasciitis. That's flesh-eating bacteria, okay? That's where we learned about this previously in the skin diseases area. So you don't want to be one of those people who is particularly susceptible to one of these more toxic versions of Streptococcus pyogenes, because necrotizing fasciitis is no joke and can be deadly, can lead to shock and even death. So this is just a depiction of some effects on hearts that is caused by inflammation associated with strep infection, particularly this would be like scarlet fever, I believe. I don't know enough about the anatomy of the heart to tell you what's going on in these pictures, and I apologize, but if you know, then maybe share with other people, because I have no idea. I don't know about hearts. So next, so with strep pyogenes, strep has surface antigens that mimic host proteins. What does that mean? That means it looks like our proteins on our cells. And so whenever we try to develop antibodies against the strep, we also can potentially form antibodies against ourself, not unlike a autoimmune disease. So that can lead to symptoms, including a lot of inflammation associated with the attempt to clear out the strep infection. There are surface antigens to protect the organism from being affected by the lysosome that would break it down when the phagocytes try to eat it up and kill it. It produces streptolysin O and streptolysin S. Those are exotoxins that injure cells and tissues. It produces erythrogenic toxin in strains that have been lysogenized. Remember, that means that we have a phage, a bacteriophage, which is a bacteria infecting virus, that its genome has become incorporated within the genome of strep. And as a result of that, now strep has the ability to produce this toxin as a result, okay? Streptococcal toxins can act as super antigens, leading to overreaction of the immune system and be particularly harmful to us. So signs and symptoms associated I don't know why I said that way. Signs and symptoms associated with scarlet fever result from, the choices are exotoxins, endotoxins, toxoids, and peptidoglycan. Those toxins that we were talking about previously with streptolysin and all of those ones, those are exotoxins, okay? Endotoxins are associated with gram-negative, and Streptococcus pyogenes is a gram-positive. Peptidoglycan is just the cell wall of bacteria, remember. Moving on to diphtheria. This is a significant cause of morbidity and mortality over time, but now that we have the vaccine, it has been controlled fairly well, not gotten rid of completely. We still have outbreaks in some areas where we don't have good vaccination availability. This is caused by the bacterium Corynebacterium diphtheriae. It is it's non-endospore forming. It is gram-positive. It does have a club shape though. It produces a sore throat, lack of appetite, fever. It makes a exotoxin, so it secretes a toxin, and is a type of toxin known as an AB toxin. They basically break apart and function because of that. Well, they have a pseudomembrane that can form in the back of the throat, on the tonsils or the pharynx, that can completely block respiration. And the Tdap vaccine, or DTaP, however you want to, the DTaP is for the babies, for the little children, and Tdap is for the booster for the older folks, anybody 11 and up. So it's included with your tetanus and your pertussis and all of that. So here is a video that we're going to watch of the AB toxin and how it works. Let's watch.
AB toxins are proteins that consist of two parts, A and B. The A portion is an enzyme that constitutes the toxic part. The B portion binds to host cell receptors. There are several mechanisms by which AB toxins enter the cell. One mechanism involves the uptake of the toxin after binding by endocytosis. After endocytosis, the contents of the vacuole become acidic, causing the A and B portions to separate. The A portion enters the cytoplasm of the cell and exerts its toxic effect, while the B portion is removed from the cell by exocytosis. Different microorganisms produce A toxins with different activities. The A toxin produced by Corynebacterium diphtheriae carries out the transfer of ADP ribose to elongation factor 2, EF2, and thereby inhibits protein synthesis. ADP ribosylation is a common action of AB toxins.
Cool. Let's go to the next thing. How about? Next is whooping cough. There are 3 phases associated with whooping cough, which is also known as pertussis. The first phase is the catarrhal phase. This is the respiratory tract infection leading to cold symptoms, just pretty mild. The paroxysmal phase is the uncontrollable coughing, which makes a whoop sound, and that can result in broken blood vessels in the eyes and even vomiting due to that. And that is referred to as the 100-day cough. It's just like you can't stop coughing. It just won't stop. You just keep coughing. You can't even breathe in. You're coughing and coughing. And then we have the convalescent phase where the bacteria will be decreasing. And you'll have a decrease in the symptoms associated with infection, but the ciliated epithelium that normally would help out with clearing out infections, that's been severely damaged and will require weeks to months of recovery. So it could leave you susceptible to other infections. So we're going to watch this part of this. I'm not all the way through, just part of this video of this poor little baby. They are the most susceptible and are still susceptible within America and in our hospital systems, particularly little newborn babies that haven't developed an immune system to the point that they can get the vaccine, and then older folks who have not kept up with the vaccine coming around them and exposing them to the pertussis. And it can be very severe for infants. So that's what this video is of. And if it is disturbing, feel free to, maybe not watch this part. So we'll just leave that there, but you can definitely get the sense of how severe the coughing can be, especially for little infants like that, who just can't fight the infection as well as, those of us who have healthy immune systems. So, oh gosh, I didn't know that was going to click on that. Okay, next slide. Okay, so whooping cough is caused by Bordetella pertussis. I'm not good at this today. It's a small gram-negative rod. It is a strict aerobe, so it has to have oxygen. And it is fastidious, meaning that it has to have extra like growth factors and stuff from its environment that it can't produce for itself. So it has something called filamentous hemagglutinin that is essential for attachment to the host's cells to, cause the infection. Then we have the pertussis toxin itself that causes massive mucus production, and the tracheal cytotoxin causing the direct destruction of the ciliated cells. All of that is just the perfect storm for just excessive, extreme amounts of coughing. The vaccine that is available, we have high coverage in the United States, keeping the incidence very low in the US, but it doesn't provide lifelong protection. So it'll wane a few years after childhood, whereas it might not be so severe for us to have pertussis. It is very, very dangerous for infants. The disease can be passed to them from adults who have not had the immunization recently. So it also seems that the organism may be evolving and the current vaccine may not provide the best protection as it used to when it was first, developed. And we also have a rise in unvaccinated individuals due to the anti-vax movement. So this is just showing incidence over time in Oklahoma and in the US. I'm not going to get into this, but it is on the rise in Oklahoma. So RSV is the next one, respiratory syncytial virus, that's RSV. Signs and symptoms of RSV include rhinitis, pharyngitis, and otitis, which is, all that's going to be nose, the throat, and the ear, those three. So more serious infections can progress towards the bronchial tree to parenchyma and cause symptoms of croup and difficulty breathing, especially in younger patients. So we have transmission through droplet contact and fomites. It's very contagious, highly contagious, and passive antibody therapy is effective in those little tiny babies and young children that can't fight the infection off well themselves, and where it's dangerous for them, we can have treatment with ribavirin as well through insulation. That is an antiviral medication in addition to passive antibody therapy. What is passive antibody therapy? I'm sure you guys know, right? Of course, it means that we are treating somebody with antibodies from somebody else who survived the disease. Okay, influenza now. We know about the flu. Influenza virus is the cause of the flu. Signs and symptoms include headache, chills, dry cough, body aches, fever, stuffy nose, and sore throat. Extreme fatigue associated with influenza infection can last for a few days up to weeks. H1N1 is the swine flu. Not all patients who have gotten infected with H1N1 had fever. Some of them had gastrointestinal distress or multi-organ system failure. It just depended on how their bodies reacted to the virus. Typically, this is very dangerous and can kill infants, elderly, and pregnant women due to their immunocompetence, and anybody else who might have immunocompetence, like people with AIDS or something like that. very dangerous. Two important factors about influenza include hemagglutinin and neuraminidase. Hemagglutinin, so that's the H in the H1N1. Neuraminidase is the N in the H1N1. And it's not only just H1N1, there's all different numbers that can go with that. So hemagglutinin, it's a molecule that has an agglutinating action on red blood cells, so it makes them stick together. That's what agglutination means. They will bind to the host cell receptors of the respiratory mucosa this way as well. So that's how the virus will stick. Neuraminidase breaks down the protective mucus coating of the respiratory tract so it can get closer into the cells, more likely to infect so that it's not getting coughed out with the mucus. It assists with viral budding as well, the release of the new virus particles. It keeps the viruses from sticking to one another, and it participates because of that hemagglutinin that could be on the surfaces. And it participates in the host cell fusion portion as well. So whenever we're going into the cell, so budding and going into the cell, and prevents things from sticking together, breaks down the mucus. Okay. Flu virus can vary in a few different ways. First, we have the antigenic drift. These are mutations of the genes, changing the amino acids. We had talked about this in the genetics chapter. If you guys can remember, that was chapter number 9 in unit 2. No, you don't remember? Okay, that makes sense to me. It was a long time ago. So when we can mutate our genes, we can have even just point mutations, which is a single base or something like that. And we have a change in our amino acids. It can change slightly the shape of the proteins or a lot. But anytime that it does change that shape, our body, how we respond to it with antibodies, and we're talking about influenza here, how we respond to the influenza, if it changes a little bit with that shape of their protein due to those mutations, we can't defend against it now with our old antibodies. So we'll have to make all new from scratch, going all the way from the beginning all over again. So every time that it changes even the littlest bit, we have to go through remaking our antibodies. So whenever our antibodies do attack them, we are in a way selecting for those that might have a little bit of change to go on to pass those mutations on. That's how we would select for newer viruses with changed surface proteins. That's a slow process, though, comparatively compared to what we're going to talk about next. That is antigenic drift, okay? The other one is antigenic shift. Big steps, okay? Drifting slowly in the wind versus shifting completely, okay? Big change. This is gene reassortment. This is co-infections. It is a rapid process. So co-infections is what's depicted down below. We have infection. There's a duck. Oh my God. There's a duck virus down here. I cannot. What are we doing? What are we doing now? Are we not going to let me draw here? No, we're not. Okay, I can't draw apparently. Okay, so we have the duck virus, which is the blue one, and we have the human influenza virus, which maybe has been floating around for a season or something. Both of them maybe wind up within a pig. That pig gets infected with both of them. They can share the large bits of the genome of the virus, which in flu, there are several different ways that they can kind of assort those things. Anyways, any of them get reassorted. We get a little bit of duck virus, we get a little bit of human virus and whatever. And that is like it's almost like a brand new flu virus to our bodies. We have never seen it before, essentially. Okay, that's how that works. Drift, small mutations due to random mutations and pressure from the antibodies that our body's attacking them with to choose the ones that can escape. And antigenic shift, which is a co-infection process, reassorting our genes with pieces of our genome in big changes with one virus and another virus making a new one. So influenza treatment, we have amantadine that will block the replication of the virus. We have Relenza and Tamiflu will block the action of the neuraminidase. Vaccination itself, we have the inactivated vaccine with 4 strains that will protect against. The attenuated one, which is the flu mist that'll go up the nose. Attenuated means it's alive, but it has been altered in a way so that it can't be virulent anymore. And then they have new recombinant vaccines that are available that can help with protecting you even without using eggs to grow a virus, which is what we typically do with the other versions. So we have to come out with a new quadrivalent vaccine each season, essentially 4 different strains of flu that we predict will be the outcome in that coming season. You have to predict it though, and it might not be the ones that come out, and there could be other changes that were introduced that you didn't predict. And unfortunately that happens a lot more than we would like. So not a perfect process. This is how Tamiflu works. Here in Tamiflu, we have the virus. You've been infected, your cells have been infected by the flu virus. That's unfortunate. Now they are trying to bud off of the cell. So let's see if I can work this pen without freaking, is it gonna let me write? Because I'm about over this. Okay. Anyways, so we have our virus that we are starting to bud out. We're assembling at the actual membrane. It will pinch off around all the contents, okay? And once it does, that virus gets stuck on the surface due to its own little its own little receptors on the surface. Good, that's my favorite. Due to its own receptors on the surface of the cell, in addition with on the virus itself. Now, normally, neuraminidase will come in and cleave that off to release the virion, but Tamiflu and similar medications will come in with their little inhibitor action and make it so that they can't cleave off that connection. So they stay stuck. If they stay stuck, they can't go and infect another cell. That's how those medications will work. In influenza viruses, minor annual variations in the antigenic makeup are called, okay, so this was either antigenic drift or shift. So this is drift. Those are the minor ones. Shift was the big step. Okay. Give you a minute to think about it because I'm sure you were. Okay, the 1918 Spanish flu. Most of us have heard about this super severe problem here. Let's go through the numbers. Yes, this ran from 1917 to 1920. It was a pandemic that affected people worldwide. 500 million people were infected across the entire planet. 28% of the world, okay, was infected. 50 to 100 million died. So that's 3% of the whole world died. 10 to 20% of those who were infected died. Now it's typically from secondary infections, bacterial infections that were opportunistic because you were infected with whatever virus, whatever. So most of the victims that did die were healthy adults. This H1N1 virus, similar to what we've seen with bird flu, caused a cytokine storm. Basically, the immune system was overreacting to this virus that it had not seen before. It was such a new strain to everybody that was overreacting to that. And that allowed for the body, since the body was overreacting to that, it couldn't do its job well to save them from the secondary infections. And, you know, back then, maybe their hygiene wasn't quite as well. I mean, look at them. They've got all these people stuck in a freaking next to each other. I don't know. That's a great idea. Everybody's sick out in the open like that, but maybe that's all they could do. I don't know. I'm trying not to judge, but I already have done it. So it's too bad. Next, we have severe acute respiratory syndrome. That's SARS. This is where COVID came from. Okay. SARS caused a high fever, what people called like a severe flu. It was spread from a coronavirus that was originally found in the civet cat. And this was in Asia, you know, it was pretty significant at the time. And it looks like a baby compared to COVID now. Next, we have Middle East respiratory syndrome, also a coronavirus. This is in camels. We've only had 2 cases in the US, but they were imported. They got sick out in the Middle East and then brought it back to the US with them. Moving on to Nipah virus or all the group being the HENIPAH viruses. This includes Nipah virus and Hendra virus. Nipah virus was the virus that the movie Contagion was based off of, one of my favorite movies. If you have not seen it, I highly recommend it. It's a great movie on epidemiology. I thought it was a fantastic movie when it came out. Now that COVID has come out, I felt like almost chills whenever things were happening the way that they were with COVID, when I went back to watch this movie again. And I was like, man, geez, like too real for me. And I think you would agree if you were to go back and watch it now. Fantastic cast, great movie. It's exciting and yeah, too real. It's too real. Yes. So anyways, Nipah virus was what that movie was based off of. It is a zoonosis, existed in, that means animals, right? Animals had it first. The fruit bat was the reservoir for Nipah virus, spread in their urine, their feces, and their saliva. In Contagion, in that movie, they called it MEV1. Not really that important, but that is the disease they called it in there, and it was based on this. Symptoms for Nipah virus in real life. This is real life, not just movie. They stole these from real life. Real life symptoms of Nipah virus infection include fever, headache, cough, abdominal pain, nausea, and vomiting in about 25% of cases. With people who have contracted Nipah virus, you will see encephalitis associated with seizure. And those, there's about 60% of them who will go comatose. Bad. It's a bad deal. So then we also have Hendra virus, which is a similar virus, but it's in horses in Australia. Fruit bats also carry that. Australia's scary. You can tell Dr. B that I said that, okay. He also hates horses. So you can tell him that, I don't know, tell him about horses. He doesn't like them. Okay, so what groups are most at risk of dying from flu? So who's it going to be? Is it going to be the infants, the immunosuppressed, the elderly, the pregnant? They're all of them, right? So that's E. Flu is bad for all of these folks. Moving into tuberculosis, right? This is an old disease, ancient, found in mummies and people have been preserved, sometimes naturally, sometimes on purpose, from the Stone Age, from ancient Egypt, from Peru. It's a prevalent cause of a disease, historically speaking. It has been referred to historically as the captain of the men of death, the white plague, and streptomycin introduction, that antibiotic, reduced rates significantly at first. Not so much the case now. So it is a re-emerging disease due to largely the HIV epidemic, people becoming more susceptible to infection and progression of the disease because of that, and the development of drug-resistant strains, a huge problem in this. That is why nearly a third of the world's population is infected with this bacterium. Primary tuberculosis, the initial infections, you will see it takes about 10 bacteria to cause, that's the infectious dose, the ID, about 10 bacteria are needed to cause an active infection. Bacteria will grow inside the alveolar macrophages. Now you're saying probably what, or you're saying what because you don't understand what I'm talking about. But if you're saying what because you do understand what I'm talking about, yes, these bacteria grow within the cells that should be fighting them. When the cells are going to come up to the bacteria to fight them, they eat them up, they think they're going to break them down. And these bacteria say, this is where I live now. And that's what happens. So they live inside of the alveolar macrophages that should be killing them and taking care of the disease. Then we have the formation of tubercles. They are granulomas with a core of tuberculosis bacteria inside of them. And the enlarged macrophages that will be harboring the tuberculosis bacteria as well. Then we have outside of that an outer wall made of fibroblasts, lymphocytes, and other macrophages trying to contain the infection, just trying to kill it off, but it's just not being successful because of just how these bacteria live within that area and how hardy they are, and the fact they live inside of the cells that are trying to kill them. That's kind of a big deal, right? That those tubercles, the lesions that they are associated with, can become necrotic and caseous, so almost cheesy-like, which is gross, but that is the truth of it. And then when they're healing over, can become calcified, so hardened. So that's a period of hidden infection, typically, where you're asymptomatic. You might have a mild fever. Within 3 to 4 weeks, the immune system will mount a cell-mediated assault. It takes a while for it to build up that defense against the bacterium, but it will happen over the course of 3 to 4 weeks. A large influx of the mononuclear cells will come into the lungs to try to fight the bacteria. That's when we see formation of tubercles. Frequently, the centers of the tubercles will break down, so they have those caseous lesions, the cheesy looking, nasty, and then whenever they are healing over, they calcify and harden. Also, at this point, you would be positive for the tuberculin reaction. And what is that? So we can see here so first of all, before we move on, we can see here in this X-ray that there is evidence of there's evidence of the development of those tubercles and maybe the rupturing of those tubercles causing inflammation and all of that, as we have the influx of the immune system into the area and fighting the area. That's going to cause what you can see on the X-ray, inflammation and evidence of thickening of the lung tissue, I guess, in areas where you have infection with the bacteria. So the tuberculin skin test has a lot of different names: the PPD test, the Mantoux test, whatever. Basically, this is the tuberculosis skin test. Okay, so we inject PPD, which is purified protein derivative of tuberculosis, okay, into the skin. You will have a little bit of bump there. We wait 48 to 72 hours. If the bump stays big or gets bigger or shows inflammation associated with it, then we consider you positive for the skin test. This is due to the reaction of the immune cells to that area. If you have a reaction, then your body has seen the bacteria before and you will test positive. Cool. So we need a bunch of stuff to diagnose you with tuberculosis. We can't just say, oh, okay, you have a positive skin test. That's not enough to say somebody has tuberculosis. Additionally, that area infection that we saw in the other X-ray, just that alone doesn't necessarily indicate tuberculosis. It could indicate other conditions. So we have a combination of things that we need to have. You will have a positive skin test for tuberculosis. You will have an X-ray that shows an area where the infection is occurring. And then we have the acid-fast test. Remember, tuberculosis is caused by the bacterium Mycobacterium tuberculosis. And what do you think when you see acid-fast? Oh, automatically you're thinking Mycobacterium. I know you are, because you're good and you know. And anyways, the Mycobacterium are acid-fast, Mycobacterium. So we can stain your sputum directly, or we can culture it if we need to. And then we can look at it under the microscope with that acid-fast stain. Remember that if you are acid-fast, that what happens is your mycolic acid that is in your cell walls or your cell membranes, your cell walls, because you are gram-positive. So the mycolic acid that's in your cell walls, that makes you all waxy and everything like that. That will stain red from the carbofuscin that we let sit on there. Quignon's carbofuscin. We didn't use heat on that one. It just sat for 5 minutes. And then we rinsed it with the acid alcohol, which was hydrochloric acid and ethanol. And then we counterstained it with the methylene blue. And if it stayed red, then you are positive. If it stained blue, then you are negative. So if it stained red, you are acid-fast. That means you are a Mycobacterium. And if you are coughing up sputum and I'm looking in your sputum and I'm seeing Mycobacterium, I'm seeing acid-fast bacteria come from your sputum, you've got tuberculosis, son. That's how that works. So we can have this thing develop called secondary or reactivation tuberculosis. Live bacteria can remain dormant and become reactivated weeks, months, or even years later whenever you have maybe a little bit of dip in your immune system. Okay, so that chronic tuberculosis, we have the tubercles filled with bacteria that expand and drain into bronchial tubes in the upper respiratory tract. We can see severe symptoms, including violent coughing, greenish or bloody sputum, low-grade fever, anorexia, weight loss, extreme fatigue, night sweats, and chest pain. That is why, ladies and gentlemen, they call it consumption, because essentially your body is consumed by the fact that you are having all of these horrible symptoms associated with having tuberculosis. Now that's secondary tuberculosis. So here is how tuberculosis is going to develop. We're going to show you pictures, little drawings of how this works. We have an area in your alveoli where you have a macrophage coming in, and it's got inside it ate up this acid-fast bacterium, right? So it's mycobacterium. It ate up the mycobacterium, the tuberculosis. It ate it up, and now it's coming into the alveolus. And it's sending out signals saying, this is bad, we got a thing in me. I need you to come and kill this thing in me. The other ones come in, but the thing in it is just reproducing, and more of them are getting spread into more and more of them, and so on and so forth. So even though these cells are showing up to try to kill this infection, they're just calling out and calling out and calling out, saying bad, 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 bad. You know, hey, there's bad here, over and over and over again. Those are cytokines, right? They keep trying to kill the bacteria, but it's not working because they're resistant, everything like that. And they're just making this big old area with more and more macrophages coming into the area that are going to become infected by those mycobacteria tuberculosis. So over time, basically the macrophages will end up dying, especially the ones in the middle, the older ones, and that will release the bacteria in the middle. That's where that caseous center will form. They need air to grow. And if they can't get air, it can, cause problems, but they don't die because they're tuberculosis. So what they're going to do is remain dormant in that area, with sometimes within those macrophages and everything, just chilling and waiting. That's whenever you get that latent disease. Okay, so they're going to just hide out there until times are good to start bumping again. So that's the idea there. And that will lead to healing of those areas, the calcification of those areas. You might think it's over, but it is not. So the mature tubercle will form and could progress that caseous center instead of calcifying for some people, or for those bacteria that are reactivating in secondary tuberculosis. That whole center will enlarge in a process known as liquefaction. It's what it kind of sounds like. We're going to start growing and releasing those bacteria in a gross way. They're just going to become gross and all of that. So the center enlarges, it becomes air filled. So now the bacteria are happy and they're growing and everything like that. And they can multiply outside of the macrophages as well. And they will get released whenever the tubercle ruptures. This will spill into the bronchiole itself and allow the bacteria to disseminate into the lungs and circulatory and lymphatic systems. Good for the bacteria, bad for you. All this damage can cause a lot of inflammation, increased inflammation all over throughout the lungs. And that's where we're going to get a lot of that sputum developing. We're going to get a lot of coughing. We're going to get a lot of blood and other discharge associated with it. So like we said, about a third of the world's population is infected with tuberculosis. It's just nuts. So when you do have reactivation tuberculosis or secondary tuberculosis, like we said, they can remain dormant for months or weeks, months or years. Your immune system will wane, and then they will come and grow back up from that lesion, the center of that lesion. Okay, that includes coughing, greenish or bloody sputum, low-grade fever, anorexia, weight loss, fatigue, night sweats, and chest pain. Like we said, consumption is what they used to call it back in the olden days. The untreated disease, if you don't get any treatment at all whatsoever, has a 60% mortality rate. Pretty crazy. When I'm sitting here telling you guys, a third of the world has this. I don't know why people aren't more worried about it. It's sort of like rabies, right? Sort of like rabies. Okay, not at all. Just to be clear, I don't want you to get confused. So extrapulmonary tuberculosis can also occur. This just means the tuberculosis bacteria have spread to other areas of the body and can include these organs. It is caused by the bacterium Mycobacterium tuberculosis, which we have brought up many times, and you should we're very familiar with this. It is a long, thin, acid-fast rod. It is a strict aerobe, must have oxygen. Its generation time is 15 to 20 hours. And it takes about 6 weeks for colonies to appear on a growth medium. And that's very slow growth there. They have something called cord factor. It's a lipid component in the mycobacterial cell wall that is associated with virulent strains, so the ones that can cause disease and present disease in people. They also have mycolic acid and laxes in their cell wall that makes them resistant to drying as well as resistant to disinfectants. Gosh, man, I'm doing bad with this clicking thing today. Click too hard. So transmission and epidemiology include transmitting through fine droplets. In this case, this is not direct contact situation. Okay, this is fine droplets of respiratory mucus suspended in the air. It can stay there for a while. Epidemiological patterns vary with living conditions around the world. Seems to be an issue with close contact living and in areas of poverty. Right, so it is an infection. It is considered to be an infection of poverty. We see increased incidence of it with inadequate nutrition, debilitation of the immune system in general, poor access to medical care, lung damage, and just genetic issues dealing with poor immune function in general. So that all makes sense. How do we treat tuberculosis? Now, I've told you previously that we have an issue with tuberculosis becoming drug resistant. And we have talked about how you get prescribed an antibiotic, you need to be taking it in full so that drug resistant bacteria don't survive. You know, you want to kill them all off. You don't want any mutants. What is going on here? You don't want any mutants, I said that weird, growing up and spreading their mutant genes around and all of that. So this is the problem. Okay. How do you treat tuberculosis? If you have active tuberculosis, the first 2 months of your treatment, now already, honey, already, I know you're probably like 2 months, the first 2 months, the first 2 months. The first 2 months of your treatment, you will take rifampin, isoniazid, and ethambutol. I don't know these words. Then after that, the next 4 to 7 months, you will be treated with rifampin and isoniazid. Now you understand why there's drug resistance. Who wants to take drugs for 9 freaking months, dude? People can't even adhere to 10 days. No wonder. And it's nobody's fault. It's just hard to adhere to taking a medication every single friggin' day for 9 months, especially in poverty-stricken countries. I mean, anywhere, really, if we're being honest. And especially considering that some of these drugs might not make you feel that great. If you had latent tuberculosis, they would treat it with isoniazid, rifampin, and rifapentine. I don't know these words, but these are the medications they would treat it with. Patient non-compliance leads to drug-resistant strains. Of course it does. Of course it does. Of course. So we have multi-drug resistant tuberculosis as well as extensively drug-resistant tuberculosis, which is worse. Okay, more resistant. Here are the areas where we see the multi-drug resistant tuberculosis that's been charted. Look at Russia and Russian countries. That's all I'm going to say. I'm not blaming. I'm just saying apparently they don't want to take their medications appropriately. So a friend of yours from Asia has tested positive for the TB skin test, but has a negative chest x-ray. What is the most likely cause of these results? Well, if you have the development of tubercles in your lungs, which is what is associated with tuberculosis, even in the primary infection, you would see that on a chest x-ray. So this person probably doesn't have tuberculosis and hasn't had tuberculosis. So I'm going to go with C, the previous BCG vaccine given. That is the tuberculosis vaccine. And their body is just reacting to the skin test because they have antibodies against tuberculosis because of the vaccine. All right, pneumonia. This is an anatomical diagnosis. It's not like a specific thing. So that means that it's associated with like the lung. It's a lung infection. That's what that means, pneumonia. Okay, so it is an inflammatory condition of the lung that has fluid filling the alveoli. It can be caused by a wide variety of microorganisms. Those organisms must be able to avoid phagocytosis or avoid being killed once they're taken up by the macrophages that would be the phagocytes. Like tuberculosis, they could live inside of the alveoli because they were resistant to the lysozyme that would normally break them down. So you either need to be able to avoid that being eaten up in general, or you will avoid being killed once you are inside of the white cells that are trying to fight you. So viral pneumonia is usually, but not always, milder than bacterial pneumonia. It's pretty common for a lot of these respiratory diseases. We saw that with the sinusitis and the colds and something else that we talked about. Oh, pharyngitis, the throat thing. Fungi can also cause pneumonia. There you go. That's a thing. We have fungi, viruses, and bacteria can cause it. Signs and symptoms of pneumonia include runny nose and congestion, headache, and fever. Usually starts out with something like a cold or something like that, and then moves into the lungs as we have collection of drainage within the lung, and then that just gets worse from there. So lung symptoms will include chest pain, fever, cough, production of discolored sputum. The patient will be pale and sickly due to pain and difficulty breathing. Severity and speed of onset of symptoms will depend on the cause, the etiological agent. So this is picture depicting what happens with pneumonia. It fills with exudate, which is fluid that your cells would be producing to try to get rid of whatever is growing in your lungs as a response to it, but it's not being as effective as we would like for it to be. It's either because in this case, we are showing pneumococci. These pneumococci are evading being the immune system because they have a capsule. Remember what we said about capsules, harder for the phagocytes to eat them up and fight them and kill them. Streptococcus pneumoniae accounts for 40% of community-acquired cases. There's two kinds of pneumonia you can have. You can have healthcare associated or community. Okay, community is not from a hospital or something like that. That's basically what that means. So 40% of the cases are going to be Streptococcus pneumoniae. 30% will just be viruses in general. And then next 20% mycoplasma, which typically cause walking pneumonia. It's not going to be quite as bedridden. 10% of the cases will be caused by these other things that are sort of random: Legionella, Haemophilus influenza, and Histoplasma capsulatum. 1%, we'll talk about those in a moment. Don't worry, we're not just going to kick them off to the curb. About 1% of hospitalized or institutionalized people will develop pneumonia. Most often, this is associated with a mechanical ventilator or endotracheal or tracheostomy tubes being shoved down in there, right? So more likely to be exposed to a bacteria from the environment. Of that 1% that has now gotten pneumonia, 30 to 50% of them will die from it. Now, things that can cause, usually caused by more than one bacteria in a healthcare associated situation, but here is a list of those that would be the most common ones to be associated with that: Pseudomonas aeruginosa, Actinobacter baumannii, Streptococcus pneumoniae, Klebsiella pneumoniae, and Staphylococcus aureus, typically MRSA in a healthcare setting. Okay, a lot of these and a lot of the stuff that we see throughout the respiratory stuff, we are working with these in the lab as your unknowns, FYI. Also, Salmonella is one of them. Okay, so other forms of pneumonia that we can see: Legionella pneumophila. Pneumophila is a bacterium associated with what they call sick building syndrome, or water cooling towers. Essentially grows within the water of those water cooling towers in those wet areas. and is spread throughout, as well as spas and ponds. That makes sense. But anywhere where you have this sort of what's supposed to be clean water area, but is not quite so clean, essentially getting moved through the building's air system, in the case of the water cooling towers anyways. And there was this incidence with elderly men. It can also be associated with smokers who have, worse lungs, getting this disease. And when it was discovered, it was at a Legionnaires conference or something. I don't know if you know what that is, but sort of like Nason's, but it's a different deal. So anyways, they call it Legionnaire's disease. It's caused by Legionella pneumophila. There's that. Then we have hantavirus pulmonary disease, spread through deer mouse feces. You're sweeping it up and you're more likely to breathe it in. That's in the southwest of Oklahoma, of the United States, okay? Then we have pneumocystis gyrovecii or carni. This is pneumonia in AIDS patients typically. And then we have histoplasma that can cause pneumonia in, typically it's deadly for AIDS patients, okay? Okay, the leading cause of community-acquired bacterial pneumonias, that was Streptococcus pneumoniae. That was about 40% of the cases. So that was that for the respiratory system. We talked about lower respiratory tract it should be mostly sterile, right? The alveoli should be sterile. And then if there's bacteria down there, they should not be long down there. Mucus and the ciliated cells will remove the microbes, especially through coughing, helping move all of that along. If you don't have the cilia, because you had maybe irritation due to pertussis, for example, that can, leave you susceptible to infection. And then we have involuntary responses that can help remove microbes. That's things like coughing and stuff like that. So, and swallowing, all of that can help remove the microbes. So that is that for the respiratory tract, and we'll see you next time for chapter 22, which I don't even remember. That is the digestive system. That's a good one. So I'll see you guys at that one.